And hello, here we go with our final video in this homework topic series. This is titled DNA, What is it good for? Absolutely everything. Maybe you recall the song reference there. Maybe you don't. It's okay. Let's go ahead and look at what DNA is good for, or more basically, what does DNA do and how does it do it? We've considered DNA as its molecular structure, talking about the A's and the T's and the C's and the G's and the phosphate and kind of how it looks. But let's focus here on what DNA actually does. DNA, in its most basic sense, is going to serve as a recipe. It's a recipe for making a product, proteins. If you want to think of the recipe being on an index card in your kitchen and a cake being the final product, the recipe is required so that you know what to do to make the cake. In very much the same way, DNA is going to be a recipe in our nucleus of our cells to tell the cells how to make all the important proteins. And again, we keep using insulin as an example, but how to make the important proteins that keep us alive. So DNA is able to act as this hereditary molecule because in its order of A's and T's and C's and G's, it spells out directions for how to make everything from your hair color to the enzymes of your body to the structures of your body. How it does it is DNA is going to make a copy of itself in an RNA form. And we're not going to dwell on this process too much. You'll see in your book, and I'll go ahead and give you these terms just so you've seen them, that there's two processes here. There's one process called transcription, and that's where DNA is made into an RNA version of itself. And then in the cytoplasm, the RNA is translated into a protein. We're not going to deal with the details of this, trust me. But I do want you to understand that DNA in the nucleus is going to make an RNA version of itself, and then the RNA version is going to be translated and made into a protein. So when I talk about RNA, what do we mean? This is another nucleic acid. Remember, DNA is deoxyribonucleic acid. RNA is ribonucleic acid, so it's in the same group, if you will. And there are differences in the two of them. DNA has a double helix. If I was going to draw DNA, for example, on this, DNA would have this structure to it. It would have a double helix, a double side to it. RNA just has one. There is a difference in a sugar and a difference in a base. And again, these are not really important differences to dwell on. What I want you to really understand, though, is RNA can be made as almost a photocopy of one of the parts of DNA, and that gives us the directions we need when the RNA goes into the cytoplasm to actually make the protein. So let's look at that process in a little more detail. Let's just consider, I guess we'll do this first, the flow of information. So from an individual, if we focus on what her cells look like, her cells, of course, are going to have DNA in the form of chromosomes. The chromosomes, when they're unwound, we see a DNA molecule. The RNA is going to be made from the DNA. You see, DNA directs the production of RNA. RNA is then going to help make the actual order of amino acids, and that's important. The protein that we get, it's a little hard to see, but these are individual dots. Those dots are actually amino acids, and there's 20 different amino acids. What DNA does is DNA tells RNA, this is the version I want you to go into the cytoplasm with, and then in a process, the translation process, the RNA is going to be read, and then the exact order of amino acids is going to be put together to make a protein, and the proteins, again, are going to be traits. In this case, brown eyes, brown hair, type AB blood, etc. Those are all basically a function of the directions that she has in her DNA. And of course, where they come from, those directions came from her parents. So genetic information flows, if you will, from DNA to RNA through these two processes. And I just mentioned these before by name, but here's the DNA. By transcription, a copy is going to be made, an RNA copy is going to be made. The RNA copy is going to leave the nucleus, go into the cytoplasm, where ribosomes, these are going to be parts of the cell, We're going to read those, translate those RNA strips, if you will, into the functional protein. So we talk about the information of DNA stored in the nucleus, but it flows, in this case it flows, from the DNA to the RNA, RNA to the protein production. So that makes this next part all the more important. And we briefly touched on this in the last video. Mutation is a change in the nucleotide sequence and the change in those bases. If an A or a T or a C or a G is deleted or added, or if a piece of the chromosome gets turned around or anything like that happens, we call that a mutation. So one question we have to ask ourselves are nucleotide changes bad? Are mutations bad? They can have a wide range of effects. They can range from no effect at all to a serious problem. Now, why these are important from an evolutionary perspective is unless there are changes to the DNA, short of sexual reproduction, there isn't any variation that's going to result. So we do need to have mutations on a small level to get some variation, but if it's a big change, as I'll show you here in a second, it can result in some pretty catastrophic issues for that protein. So what might happen if we change the sequence? Kind of depends. Let's take for a second and just see how nucleotide sequences can get changed. Again, mutations in DNA can change the protein that's produced. Can change the protein that's produced. But how does DNA get changed? They can be spontaneous mutations. That is, whenever we go through the process of copying our DNA, we may not do it perfectly. And if we don't do it perfectly, then we can introduce a small mistake into our DNA. It might be caught, it might not be, it might have no consequence, could be serious. But these are what we refer to as the spontaneous mutations because these are not caused or induced by anything. They're just going to happen as part of the cell processes. But there are ones that are going to be induced by mutagens. If we're exposed to the radiation, if we're exposed to the cancer-causing products of cigarettes, even chemicals in our foods and radiation from the sun can all cause small changes in the DNA. Does that mean that you're going to get cancer if you eat a cheeseburger? Does it mean cancer if you smoke a single cigarette? No, not saying that. But you can have cells that get damaged, and over time, the damaged cells can produce more damaged cells. And as we said in the last video, if those damaged cells actually, or those damaged sequences rather, are genes that control like the rate of cell reproduction, that can result in cancer. So mutations can be bad. But I keep stressing this, do they have to be? Well, it's helpful to understand how DNA is read. If you ignore the red lines for a second here, you can see in the DNA TAC, GGT, AGT, CAA, ACT. That's how DNA is read. There's no punctuation in DNA. There's no capitalization. This is just the order of those bases: thymine, adenine, cytosine, guanine, thymine, etc. Those are the bases that go down one side of DNA. Don't worry about the messenger RNA sequence for now, but and you might see again, here's uracil. This is one of those different bases, one of those differences of RNA and DNA. But when DNA is going to be, and you know the word here, when we actually go through the process of transcribing DNA, this is the RNA version of that. And then this RNA version is going to be translated into the sequence of amino acids: methionine, proline, serine, valine, and there actually is a stop sequence in the DNA that says that's it, that's the end of your protein. What I want to show you here, though, while we don't stress, is that DNA is read in these three-letter words every time, and every time. So DNA is going to be transcribed into messenger RNA, translated into amino acids, and because we have TAC, we have these three letters mean this. Then when these three letters are translated, they mean this. GGT, this one is going to be transcribed to here, translated into that amino acid. So it's important to see it's three-letter chunks, and we call these chunks, let me give this term, we call these chunks codons. So since DNA is read in 3-letter chunks into codons, then that's really important if we have a mutation happen. Here's what I mean. Let's go look at this one. Let's say we have, this is our sequence. ACA is going to be transcribed to UGU, and then it's going to be translated to the amino acid cysteine. Maybe it helps if you see this. The cat saw the hat. Okay, notice how I have these 3-letter words. I could have a mutation. Something could happen to this original sequence where the A, where this A gets mistakenly put, a G gets put into its place. And now I have a CG, which gets turned into the RNA version UGC. Notice in this case, it did not change the amino acid. There is some duplication for some of these, so that would be no issue. And that's kind of like this. If I had a mutation where I changed this to a lowercase a, just play along. The cat saw the hat. We didn't change the meaning at all of that particular mutation. So that's called a silent mutation. The DNA got changed, caused a change in the RNA, but didn't change the amino acid, so nothing happened as far as we can tell. A missense error is when we change something that does result in an amino acid sequence change. So here the A got changed to a T. So when we transcribed this, we got a different sequence. Here you see AGU, where before it was UGU. Well, AGU means serine. It means that amino acid. So I'm showing you here kind of how a word got changed. The bat instead of the cat. Okay, that's a missense mutation. Is that a serious mutation? Did I seriously change the word? No, but I changed it. And that might be quote unquote okay. It could be a problem, but it might be okay. A nonsense mutation is when we put in one of those stop sequences by mistake, and I just put in a period here to represent the stop sequence, the catsa. And if that's all you knew, you don't know anything else. If this was an amino acid sequence and we had just that, that would be a protein that would fold up and be the wrong shape and wouldn't work. So nonsense mutations are pretty serious. So as you see here, a point mutation where we change a single letter can have varying effects. It can have no effect, like a silent mutation. It can have some effect, like a missense mutation. It can have a great effect, like a nonsense mutation. Let me just show you one more kind of mutations here, and then we'll wrap up here. This is called a frame shift mutation. If I were to add any letters, add any bases into my sequence, or take them out, is that going to be a more serious error or more serious issue than we have with point mutations? Again, here's our sequence. Here's our starting sequence. Let me grab a pencil. So here's our DNA. It gets transcribed to this version of RNA, which gets translated to methionine, serine, cysteine, leucine, and arginine. Great. Okay, here, because of either a mutagen or because of a spontaneous mutation, we insert an A. It kind of gets put into the place here. And now because of that, now we actually have a different amino acid in this second position. Because we now have to shift everything over, and now we have to read these three, kind of hard to see here, then we have to read these three, and we have to read these three, so everything got shifted over a spot, and now look we get, we had methionine, serine, cysteine, leucine, arginine, now we have methionine, leucine, proline, 3. Boy, that's a bad mutation. That's a big issue because we've really changed that. So if you get a mutation like that, a frame shift, it's going to throw you off one slot or even 2 slots, and you're going to end up with a really different kind of protein. So frame shift mutations often result in defective proteins. What I have here, for using the same sentence as before, the cat sat, would now that this loses all meaning and all understanding. So what I want you to take away from this is DNA is a really, 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 really important recipe set. And that recipe set is going to be transcribed and then translated to make a protein. The protein is the goal. However, if the mistakes occur in the DNA, either through spontaneous mechanisms or through mutagens, and we change the sequence, we may not, but we really could change the resulting protein. So that wraps up this topic on DNA. There's going to be an assignment following. If you need to go back and look at this one's a little bit involved. You may want to go back and look at this video one more time. That does it. Have a nice day.